In order to discover which codons correlated to which amino acids, Nirenberg conducted two separate experiments that led to the discovery of the coding dictionary. The first experiment was conducted by Nirenberg and Mattaei. What they did was use a poly-U homopolymer, which is a long strand of uracil bases, a cell-free cell extract, which is a cell not bonded to its cellular membrane, 19 amino acids that were not radioactive, and 1 amino acid that was. For the experimental process, they placed all of the components of the experiment in a tube. Then, after this, they incubated it and waited for it to translate. They then filtered the protein and observed if it was radioactive. This radioactivity would indicate which amino acid the particular RNA sequence would code for. This experiment was repeated 20 times, with each time a different protein being radioactive. When it came to poly-U, it was found that phenylalanine was the only amino acid that turned out to be radioactive as a response. This meant that the RNA sequence UUU coded for PHE, or phenylalanine. In order to ensure that these results could possibly work for other sequences, the scientific pair repeated the same process with the poly-A and poly-C and poly-G sequence. Poly-G showcased no results. However, just as with poly-U, the poly-A and poly-C found their amino acid pairs of lysine and proline, respectively. Now that Nirenberg had discovered this much about RNA sequences and amino acids, he wanted to expand his discovery beyond synonymous RNA sequences like UUU, AAA, and CCC. In his next experiment, he wanted to discover which amino acids correlated with codons that had differing bases. In Nirenberg and Later's experiment, they used a trinucleotide codon, 20 tRNA with amino acids attached, and ribosomes. Just like the first experiment, they made sure to have one radioactive amino acid which was attached to a tRNA. After placing all components into a test tube, they waited for any reactions, also known as binding, to occur between the codon, tRNA, and ribosome. Once this period was over, they filtered out the solution and inspected what the filter had caught. By using this method, they intended to catch any tRNA ribosome pairs that were able to bind, and the ones that did not bind were able to fall through. This is where the radioactive component of the experiment came in as well. When Nirenberg and Later inspected the filter paper, any trace of radioactivity would indicate that the particular RNA sequence allowed the amino acid attached to the tRNA to be expressed. This in turn meant that the specific pair would be added to the coding dictionary.